Hello everyone, welcome back to the Meet channel. Today I would like to share a very interesting talk that I have delivered as an invited speaker in an international conference on current trends in waste management, reuse and valorization, which has been organized by Sandeep University, Nasik in association with Society for Green Environment, New Delhi on 26 February 2022. I, Dr. Dipankar Ghosh, Assistant Professor, Department of Biotechnology, is honoring the lab Microbial Engineering and Algal Biotechnology Laboratory at JIS University. Today, the topic that I'm going to deliver and share with you is systems biology approach to screen and identify algal phosphatases for detecting heavy metals, which could be a sustainable platform in near future. So this is the specific orientation, the way I would like to proceed, followed by introduction, rationale, following experimental design, result discussion, conclusion with some further reading. Heavy metals have enormous toxicity on microbial metabolic pathways. Its corresponding enzymes, indiscriminate disposals of the heavy metals in nature, has a huge detrimental and bad impact on environment and ecosystem, as well as on human health. Thus, the minimal concentration, minute quantity of heavy metals should be maintained in the environment with proper real-time monitoring. There are broad spectrum of approaches to exist that will allow to screen and detect the heavy metal contamination in the ecosystem at current scenario. However, these current approaches suffers due to the lower electron selectivity, inaccurate response, and higher cost involvement. Moreover, current avenues are not directly consolidated with toxicity impacts of those heavy metals. In that particular scenario and rationale, algal phosphatase enzyme transforms the phosphodiesterase molecule to get degraded into solubilized inorganic phosphate moiety. So its catalytic activity has strongly been inhibited in presence of even minute amount of heavy metals. Thus, particular feature of the algal phosphatase that will allow to detect these toxic heavy metals, those who are existing in nature or in the human stream, even though they are present in a very minute and tiny quantity. So, thus the present study will help to find out the novel algal-based alkaline putative phosphatase protein sequences and needs to get performed some bioinformatic systems phylogenetic studies before in silico characterization or featureization, followed by algal-based alkaline phosphatase enzyme affinity towards diverse heavy metals, especially cadmium and mercury. So based on this rationale and scenario that we have been briefly outlined, the current study is dealing with on the in silico screening and identification of algae-based alkaline phosphatases following systems biology approaches. Now let's move on to the experimental segment. So there are certain distinct steps which has been depicted over here as a very nice and brief outline and overview. It will begins with the diverse ranges of algal phosphatase retrievation. That is, the diverse range of algal phosphatases. Some things are known, some of them are putative protein sequences have been retrieved from the KEGG or the UniProt database. Retrieved protein sequences have been undergone multiple sequence alignment, followed by phylogenetic studies using Cluster-W algorithm, and molecular evolutionary genetic analysis has been done following NJ algorithm. Putative protein 3D homology modeling has also been generated using EADOC, considering the most similar protein models which are already present in the PDB database as a salt crystal structure of those enzymes. Putative protein homology model then needs to get quantified and quality needs to be checked, evaluated through the Ramachandran plot, global model quality estimation, and mole probability score. Afterwards, the transmembrane helices in protein sequences have been predicted to make sure which enzymes are going to get secreted outside the cell naturally. Finally, the putative protein model have been docked with the two selected heavy metals. One is cadmium, followed by mercury, to predict the metal affinity and the microorganism or algae-based amino acid residue involvement in those interactions using the MIB algorithm. So based on this all experimental workflow, we come up with lots of predicted results, and we will be follow up the discussion on that. So homology modeling using EADOC has shown that the most algae-based alkaline phosphatase model exists in favored region. If you consider the Ramachandran plot, an average value which has been observed higher than 82.5%. Based on this favorability, phylogenetic studies have been shown and performed that the eukaryotic algae and the prokaryotic algae, they do exist in the different clades based on their very conserved domain and motif in their catalytic site, which are supposed to get interacts with the different forms of heavy metals. Even they do exist in nature in a minute quantity. The metal ion binding also plays a very important role. That has been followed here as an in silico approach, have clearly shown that there are certain algal phosphatases are most predominant that can be used as a biosensor enzyme to screen the cadmium heavy metal toxicity in environmental sample in different habitats. In the contrary, there are certain other eukaryotic algae alkaline phosphatases, they do exist and predominantly can be used as a biosensor enzymes to identify mercury toxicity in the environmental sample. And that will allow you to understand the enzymatic affinity towards different enzymes, depending on the MIB score for mercury and cadmium respectively. Afterwards, to check whether those enzymes are actually get secreted outside the cell, needs to carry out the MHHM analysis. That has been the most of the algal alkaline phosphatase. It shows they do have a secretory in nature, excluding certain eukaryotic or cyanobacterial alkaline phosphatases. So based on this very extensive study, which has been done on the in silico platform, preliminary in silico prediction clearly shown that there are certain alkaline phosphatase that can be used that will allow you to screen the cadmium heavy metal toxicity, including the mercury toxicity, those who are present in the environmental sample, different reservoir or habitat in ecosystem, even though they exist in a very minute quantity. However, TMHHM, transmembrane helix analysis, also depicts the presence of transmembrane domain in the AP, where the transmembrane segment may cause slow or no release of enzyme from the algal species, from their cytosol to the exterior environment. Now we come to a future outlook section. However, proper in vitro or in vivo validation experiment needs to be performed, because whatever we have shown here, these are the prediction models, those who are based on different in silico systems biology approaches, bioinformatic tool applications, but it needs to be get validated by in vitro and in vivo experimentation. Putative protein PDB model that we have identified that needs to be undergo for crystal structure development, followed by dynamic simulation analysis, development of trajectories as a future upgradation of this particular study to ameliorate the protein model accuracy and for better data interpretation. So this is like a paper we have published in Springer Nature last year that have been explaining in this particular lecture talk. So if you are interested, you can go for further study and get a lot of other glimpses and essence of this work. Finally, I would like to acknowledge Dr. Pankaj Chaudhary, who is the President of Society of Green Environment, and for inviting me for this very great platform, by sharing this great platform. I would also like to thank organizing committee of Sandeep University, Nasik, and Society of Green Environment, New Delhi. Would also like to acknowledge Department of Biosciences, JIS University, and the high officials, those who allow me to pursue this lecture for building up the collaboration and the crosstalk on the base of science and technology, and all participants, those who are here and enjoying this session. I hope you enjoy this story and you get inspired. This is my small group. I've been working at JIS University, and the students have been helping me out a lot. Their supports are really appreciated. PhD scholars, MSc research project scholars, and MSc interns. I would like to also announce one thing. This particular presentation recording will also be available in the MEEP Sciences channel that we have been operating on the behalf of our department, on the behalf of our laboratory. Thank you very much for your great attention and participation and to be with me. Thank you very much.